let's get started. Uh, so today our topics are mainly Boolean models. We'll go through Boolean models, how those work, and how you can apply them. We'll look at a few examples as well, and also we'll be talking about robustness in the end. But primarily, primarily base topic is Boolean models, and we'll go through them. Okay, so let's get started. First, just to recap, gene regulatory network, we have studied this before. Just to recap this. So, what is a GRN? It is basically a network of uh, primarily transcription factors and DNA cis regulatory elements called trees, along with various genes, and how these various elements control the expression or regulate the expression of that particular gene. That interaction is shown in gene regulatory network. So at the forefront of gene regulation are transcription factors which interact with specific regions of the DNA called cis regulatory elements known as T. And these regions can be promoters or enhancers or other upstream elements of the DNA of the gene that has regulatory mechanisms. Basically, a transcription factor or another protein can go and bind to that area and regulate the expression of the gene. Uh, Reconstruction of these networks poses significant challenges that necessitate the development of powerful and efficient tools to unravel the regulatory interactions of GRNs. But today we'll look at GRN from a modeling point of view, not from a reconstruction point of view. We have seen that. So let's get to it. So first concept that comes here is basically logical models. What are logical models? These are the most simplest of the models you can think of, the most basic and the simplistic modeling methodology, and it is discrete and logic based. So you can either have discrete values, so 0 or 1, either it is off or on, not continuous values like we see in case of say ODE based modeling. We assume continuous parameters, but here the values are basically the values are discrete. It can either be off or on. There can be nothing in between, which is not actually in line with biological systems, as we all know, because biological systems show this spectrum of states where it can move from one state to another, but like those are not two ends of the spectrum. There can be like middle states that are between these two ends. There can be other states, statal states within the spectrum, which can be equally useful for the system to be in. But in this case, we will not assume those kind of transitions, because we'll assume either a 0 or a 1. Either it is fully off or it is fully on, not partially off or partially on. But in case of in real life biological models, it is not true. The assumptions that we'll take here are not always true. So the reconstruction of this regulatory network that controls the development of sea urchin embryos is an example of profound insights that quantitative examination of regulatory models can provide. The work demonstrates how maternal cues initiate at the activity of the regulatory network and how this network orchestrates developmental process. This is in case of developmental process of sea urchin. In certain animals or certain lower animals, not higher vertebrates. So in certain lower animals like sea urchin, also you'll see in cases of say for example in insects, in Drosophila. So this maternal cues, the signaling that is uh, the the signaling component that is coming from the maternal uh, part of the embryo, that is very important in uh, regulating which side will be in the embryo, which side will be the anterior, which side will be the posterior. So the maternal signaling is very important in case of development process of lower organisms. In higher organisms, it is much more complex. So I am not going to that. But uh, basically, in this study, in the paper, what they did was they studied this whole regulatory network that controls this the developmental processes of the sea urchin embryos, and they found out this maternal cues that initiate the activity. This was kind of a validation that of the knowledge we already had about the developmental processes. But this is a very good example on how you can use these kind of logical models to predict certain things. So in this case, it was which signaling is more important for the whole regulation of the development process of the employee. So logical models represent the local state of each entity in a system. For that can be genes, that can be proteins, that can be small molecules, or that can be other things as well. That can be, say, for example, a state. So you can also, so these are your, if you're talking about genes, proteins, small molecules, these are at a microscopic level inside the cell. But you can model the whole cell as well. So the basically you can model the state of the cell. So it can be either it's in state one or state zero. So one example I'll give you is basically the differentiation of T cells. So T cells, what happens is basically TH0 is basically the initiator helper cells, or you can, they're not stem cells exactly, but they're more evolved than stem cells, but those are initiator helper cells, those TH0 cells, and those divide into uh, TH1, meaning depending on the infection, depending on your adaptive immunity, when it is required, those TH0 cell will divide into TH1, TH2, and TH17, which in turn will secrete different cytokines. So TH1 will secrete a cytokine, TH2 and TH3, depending on your need. This differentiation occurs. So these cell states also you can model as logical gates. So for example, you can consider this TH0 as a zero state or TH1 as a one state. So the cell can either be in TH1 or TH0, or you can assume that the zero, like basically in TH0 the cell state is zero, or in any of TH1, TH2, and TH17 the cell state is one. So the cell can either be in a TH0 state, or it can be either in TH1 or TH2 or TH17 state. So either in zero or one. So you can also model cell states, not only microscopic elements like genes and proteins. You can also model cell states on higher level. Also, you can even model certain tissue expression levels or tissue states. You can say for example, if it is a tumor, it is one. If it is not, it is zero. So what happens is, the more higher you go up in organization, the more noise that comes in your data, the more noise comes in your model. So it is very hard, the model that will uh, properly explain the gene on and off mechanism will not maybe properly explain the cell state transitions, because this is happening on a macroscopic level, and the genes and the proteins, the model for that is on a microscopic level. So it is imperative that you'll lose some information. One second. Huh. So it is imperative that you lose some information. So here also comes the fact that, like, you are only considering a zero, off or a on state. You are not considering any other states in between that. So there's a lots of information that gets accumulated the more you move up this organizational order. So at the gene or a protein level, the model can still be implied because it is at a microscopic level. The more you go up and up and up, say from genes and proteins, you're going to cell, then you're going to tissue, then you're going to some organizational state, anything, the more you go up and up and up, the more assumptions that you make about this zero and one, this off and on, this the error gets accumulated over the organizations. So it is much more difficult to model cell states using logical models than uh, genes, because it is in biological systems, the whole system is very heterogeneous. So one cell state you cannot define. Say for a tissue, you cannot say it is all the cells are in the same cells. So there, I'm digressing, but the point is, it is very hard. The more higher up you go, the harder it becomes to model. But if you make certain assumptions, like in this case we are making this assumption, but in those cases, if you make certain assumptions, then obviously your model will work. But you have to make those informed and educated assumptions. So entity levels are updated so this model goes on, and entity levels are updated at each step according to the regulatory functions given in this figure. So this is basically a Boolean network. So the three entities are there, A, B, and C, which can either exist in 0 and or 1. And these are transitioning according to the regulation functions here given here. So basically, B is you can say B is an OR function between A and C. A is 0, C is 1, B is 1. So like this, these are regulatory functions that is basically imposed on your model, imposed on your system, and the states of this of the genes or the proteins you're modeling, the change of states of these particular elements is governed by these regulatory functions. So in this case, the system progresses through, if you see, according to these regulatory functions, the system progresses to six different basically stages, and this is what it forms is basically a trajectory. Okay, like you said, what it, meaning if you model this, say if you have a three gene network and these are your regulatory elements, and if you model this, what information are you getting? So the primary information you're getting is what are the stable states of this C. What are the stable states this particular three node network can be in. So in this case, these are six global states. So this kind of information you can get from using models like this, and uh, using regulatory functions on how these genes or proteins are expressed. This is another type of logical model. This is known as what is known as PetriNet. We'll not talk about this much, but yeah, this is another kind of logical model. This consists of basically entities and transitions, and these are connected by arcs. This kind of you can imagine kind of a hypergraph situation here. So the transitions consume tokens from input places and produces token in output places upon firing. And this transition activation occurs when input places contain sufficient tokens. This transition T1 and T3 here can alternate indefinitely, because like both are very stable states, and the regulation is such that it can alternate indefinitely. But whenever T2 is activated here, it restricts further firing. So these are different kind of. So imagine this in a scenario of yeast metabolism or E. coli metabolism. So from these kind of networks or these kind of models, you can claim like if a certain basically a transition is happening already. So for example, this T2, if this T2 in this case is happening, then the system does not fire anymore. So these kind of questions you can answer. Say for example, if this particular metabolite is formed, then maybe the metabolic pathway will not go into the pentose phosphate pathway, or it will not go into that gluconeogenesis pathway. These kind of inferences you can draw from logical models. Okay, moving on. So this will study extensively only Boolean models. So what happens is quantitative methods like using ODEs and stochastic simulation. These are very good for studying cell dynamics, because you're modeling the whole basically you're modeling the states of the cell or the protein or the gene across time. So it is for studying dynamics, it is very good using ODEs and stochastic simulation. But your mechanistic knowledge and your kinetic parameters must be available. Without that, you are not able. But due to the inherent noise that is in biological data, making precise parameter estimation is challenging. See, we studied about parameter estimation, different methods you can use to estimate the parameters. The thing is, along with the data, as your data increases, your parameter estimation difficulty also increases multiple times. So often it is not even feasible to make those estimations. In certain times, it is impossible. So also, these models can be computationally demanding, and in some cases, if you have a big enough network, it is possible that your you do not have enough computational power to model the whole network, or even if you have enough power, the time taken to model such a network is basically not practical. So these are kind of limitations that you can have with dynamic modeling using ODEs and stochastic simulation. So an alternative to that is what are Boolean models, which are qualitative models. So these are preferred due to their ability to offer insights into system dynamics in a reasonable time frame, because there are not too many simulations you're doing. You're assuming a lot of things, including that 0 and 1 state. So it becomes much more simpler to simulate networks like this and and generate results, because uh, it is much more simpler than ODEs and stochastic simulations. So Boolean networks are common qualitative modeling framework for gene-regulatory networks, and these are capable of describing various biological phenomena such as oscillations, multi-stationary events, switch-like behavior. Boolean networks are powerful tools for studying GRN dynamics as well. So in a Boolean network, as I said already, the Boolean variables represent, we are talking about gene regulatory networks in this case. So the Boolean variables represent gene activation states. So 1 for active, 0 for inactive. And the Boolean functions will determine the state of each variable based on the states of other variables. So basically, you'll have relationships of each gene. So A is not activated when C is there. So A equal to not C. So these relationships, they define how the dynamics will be for the expression, and the value of the Boolean value of one expression of one gene is dependent on the value based on the states of other variables as well. So like the expression, the example like here, A not equal to C, A equal to not C. So uh, A is dependent on the expression of C. So relationships, these relationships are very well modeled in Boolean network. So you need to, when you are making this kind of models, when you are uh, using these kind of models to the model say any kind of gene related network, the most important thing that you should know for this to work is how are the relationships between individual elements of that network. So you need to have a good, what to say, like you need to do good literature review or literature survey to understand what are, is this particular gene antagonistic to the other gene, or is this particular gene protagonistic to the other gene, or do they have no relationship at all. So these kind of information you cannot get from computational studies. At least you have to do wet lab experiments. So what you do is there have been many such experiments using many such genes. So what you do is you do literature survey and find these relationships. But these relationships are very important if you're simulating a Boolean network model. Uh, Boolean functions can be represented as logic formulas or truth tables. We'll see like examples of this. And graphical representation of Boolean networks is directed graphs, because you need to know the relationship is inhibiting, is activating, whatever it is. So the nodes represent the Boolean variables, which are the genes in this case of the gene regulatory networks. The edges will denote the influence of one variable with another, with two types. One is promotion, which is positive in influence, which is more activation, and and the other is inhibition, which is the negative influence. So either one gene can activate another gene, or the gene can inhibit another gene. And promotion and inhibition edges are typically distinguished using the different arrows. So this is basically a representation, Boolean representation of network. So this is a network having four genes, A, B, C, and X, where A and B have a positive influence on X, and C has a negative influence on the same. So basically, if you see this model, this panel, so there are four genes, A, B, C, and X. A and B are activating X, and C is inhibiting X. This suggests that in the formula expressing the condition for the activation of X, there will be a positive occurrence of A and B, and there will be a negative occurrence of C, which is completely intuitive, because your A and B will be basically positively influencing, and C will be negatively influencing. So it is evident that it will the formula in which the condition will be expressed for the activation of X, it will have A and B as positive and C as negative. This is because the truth of A and B favors the truth of X, while the truth of C disfavors the truth of X, which is basically equation. However, this one thing you notice that this representation is very abstract. It does not specify the way genes influence each other. So it is basically we know the relationship between A, B, and C. We know the relationship between how A and B and C are acting on X, but we do not know how these are A, B, C themselves are connected with each other. So this is something that you'll want to know if you are doing certain Boolean models. So yeah. Now it's basically representation of these Boolean relationships is done in this way. So, say for example, but one thing you notice, this is redundant. So, for example, I am seeing this network A, B, and C. A and B are activating X, and C is inhibiting X. So, see the first equation A and B and not C. This is a perfectly good explanation for this graph. If you see this graph, and if you're told to write the Boolean function, a Boolean relationship between A, B, and C and X, this is a perfectly all right relationship. Same in the case of the second and the third. So second is either A or B and not C, which can be true from this image. And the third one is A and not C, A and not C, or B, or anything either of uh, these three, or like there can be much more. So any of these relationships can be used to explain this basically graph. So this what I'm trying to say is basically the representation is very abstract. So this doesn't give us much information about how they are like how these three genes are like related. So A, B, and C, how these three are regulating themselves, or how the dynamics of these three are changing depending on each other. That information is not present here. So this is what I'm trying to say. So this is an abstract. This is a very abstract Boolean relationship. So you have to define your relationships very well. This kind of abstract relationships won't work because there can be multiple possibilities. So you need to define your Boolean relationships. In the previous Boolean functions, A and B have a positive influence on X, while C has a negative influence on X. As a consequence, several Boolean networks can correspond to the same graphical representation. As you see, like multiple relationships can be used to explain that same graphical representation. On the other hand, starting from a generic Boolean function, it is not always possible to obtain a clean and simple graphical representation. As it is true, like from this graphical representation, it is very hard to get a single Boolean relationship. The same is true for the other way as well. So if you have a Boolean relationship, it is not always possible to have a simple graphical representation. So both are true. While it is easy to easy to do it for formulas in which occurrences of each gene are either all positive or all negative. For formulas containing a gene having both positive and negative occurrences, the translation into graphical representation, it is not straightforward, which is this case. This, for example, happens in the second case. So if you see the second equation, in which A either has a positive or a negative influence on X, depending on the activation states of B and C. So, like if B is activated and C is not activated, uh, A when like it is very hard to define the relationship between A and X from the second equation. It is dependent on the relationship between B and C, right? Depending on the activation states between B and C. This cannot be trivially represented in a graphical form because the type of arc connecting A with X cannot be defined. Because from the second equation, it is not clear what is the relationship with X with A, direct relationship. So A possible solution to create an extended is to create an extended graph which includes nodes representing combinations of genes rather than simple genes. So this is one technique that people use. So one node, generally we use it as to represent one gene. So in these kind of cases, what they do is they extend the graph and include nodes which are basically not a single gene, they are combination of genes. That can be a pathway module, that can be any kind of basically genes that you want. So this is one problem that you have to keep in mind while translating from graph-based networks to Boolean relationships. So the one-to-one mapping should be there. So your relationship should be well defined, and it should be clear what is the relationship between the genes. Okay. Next, we come to the concept of synchronous and asynchronous dynamics. So, what is this? Basically, in layman terms, synchronous dynamics assumes that after this basically the gene is activated, the next step is occurs within the same time frame, or the time differences between the two processes is very low. So it is, you can assume it to be a same time frame. And in asynchronous, it is kind of opposite. So your activation can happen after a certain amount of time. That like that information you can model using asynchronous dynamics, which is not possible in synchronous dynamics, where they assume that all the activation is done in one frame, or the one time frame, or all the inhibition is done in one time frame. So the relationship between two genes, that the outcome of it is within that particular time frame. So the dynamics of Boolean network model is given as a sequence of steps in which the value of the Boolean variable are updated with the values obtained by associated Boolean functions. Usually, two alternative updating schemes can be considered, which are known as synchronous and asynchronous. The synchronous scheme is deterministic. All the variables are updated simultaneously according to the associated Boolean function. So, the simultaneously is important here because you are assuming it is all within one time. This approach based on discrete time steps is computationally efficient and easy to implement. Now, synchronization of updates implicitly assumes that all biological events have the same duration. This is often a quite strong assumption from a biological viewpoint, since in general the timescales of biological events can vary from fractions of seconds to even hours. So this is something that you need to keep in mind. To address the problem, we can make the use of asynchronous screen, which assumes that only one variable can be updated in a single step, and every variable is equally likely to be updated. And in this approach, the nodes can be updated in any order, and the dynamics becomes non-deterministic. So the major difference between synchronous and asynchronous is you update all the parameters update all the Boolean functions in one step simultaneously for all the genes that you do. In case of synchronous, the underlying assumption is that the biological events have the same duration. In asynchronous, this is kind of the opposite. You can update the variable. You can update one variable in one step, and each of those variables are likely to be are equally likely to be updated. So basically, it's a random variable. You just pick whatever the probability is. With that probability, you update. In this approach, the nodes can be updated in any order, and the dynamics become non-deterministic. Okay, so moving on. So next, we come to what are known as attractors or attractor analysis. So given an initial configuration of the Boolean variables, the model evolves following either a synchronous or an asynchronous updating scheme. This causes the values of the variables to change, describing new configurations of state reached by the model's algorithm. After a number of steps, the model typically reaches a state which is known as a sequence of states. It can be a single state or a sequence of states. This is what is known as an attractor. The attractor can be one of the three types. One is self loop, which is a single state attractor, which is a simple loop, which is basically cyclic sequence of states, and the other one is a complex loop, which is alternation between two or more simple loops. So they can be self, simple, or complex. So when the synchronous update scheme is adopted, it is only possible to reach self loops and simple loops since the dynamics is deterministic. In asynchronous case instead, it is also possible to reach complex loops. The simplest type of attractor, the self loop, corresponds to a fixed point in a Boolean function of the networks. All the updates do not change the values of the variables. Since the Boolean functions are computed independently from each other, the self loop is the same irrespective of the chosen values of the variable, and irrespective of the chosen updating scheme, which is either synchronous or asynchronous. So since the Boolean variables are independently computed, so the self loop is a very good strategy, because if the Boolean variables are computed independently, the self loop is independent of the updating scheme, which is that can be either synchronous or asynchronous, which is a very important feature people use. Moreover, in complex networks, the update schemes influence the probability to reach the same attractors, but the attractors reached in the synchronous model are not present in the corresponding asynchronous model. So, in complex network, what can happen is when you update using synchronous or asynchronous schemes, the update schemes generally influence the probability to reach the same attractors. Which are the attractors? The attractors are the same ones that are reached in the synchronous model, but not present in the asynchronous model. It's kind of, it is present in synchronous, not present in asynchronous, or it can be vice versa as well. So in complex network, this is an issue because this will influence the probability to reach that attractor. So it will depend on. So our idea is simple loop. In simple loop, it is independent of the updating scheme, right? But in complex networks, what can happen is even if you're using a simple loop strategy, the updating has some influence on the probability to reach the same attractor. So there can be certain bias of the system to go towards a certain attractor and not towards the other, even though the other attractor is maybe more feasible for the state. The identification of attractors is biologically relevant because they are often correlated to the gene activation configurations of specific cellular phenotypes. Besides the analysis of attractors also help in examining the system dynamics and comparing with experimental data. This is what we are trying to do. Ultimately, our goal is to see not all of the time dynamics, but basically we have a certain system prediction and we want to check with experimental detecting the attractors is sometimes challenging, in particular when the exact initial configurations are unknown or when large Boolean models are studied. And as in regards to the first problem, which is basically the exact initial configurations are unknown, it requires testing the model by considering possible 2 to the power n initial configurations, where n is the number of variables in the. An approach to overcome this problem is to sample a large number of initial configurations and to calculate the probability to reach certain attractors. We'll see how these attractors work in that example, but yeah, this is the general idea of what the attractors are. Okay, now we move into one example, which is the bonding of the human and the Mycobacterium tuberculosis virus. And basically, this virus causes tuberculosis. And what this study has done is they have shown the interaction of this of the various components of this Mycobacterium along with various by components I mean genes or pathways or proteins. So uh, various components of the mycobacterium and along with the components of the human. So there is often crosstalk between the human genes, proteins, and different elements, along with the bacterial protein, bacterial gene. This kind of crosstalk is very prevalent, and with the help of this crosstalk, the bacteria actually adapts itself to this environment because it is a new environment for the tuberculosis bacteria. So using these crosstalks and all, they evolve inside the host. So uh, the first step to modeling the human MTB interactions is to get the network. So how do you construct the network? Uh, you use literature data or literature survey or literature review, and you check for how different networks are how different pathways are studied, and how they are interacting with the bacterial side of the pathways, and how they are influencing the human side of the genes as well. So these kind of information you need to find for extensive literature. Uh, After that, for this particular model, it also incorporated both adaptive and innate immune responses, as well as various aspects of this mycobacterial virulence and defense mechanisms. Obviously, when a bacteria will infect a human, so its immune system will be activated. So there will be also crosstalk between immune cells and the bacterial components. That can be the bacterial cell itself, that can be the bacterial genes or pathways, or any kind of proteins that it is. So you have to incorporate those information as well. In this case, they have done. So what it is trying to do is it is aiming to provide a systems level understanding of these interactions and addressing crucial questions regarding the immune response factors and bacterial defense mechanisms. Because if you can model the crosstalk, it is very like it can be very useful to understand which components of the bacterial network are important in this infection, and how we can control that, and how we can basically counteract that, how we can make drugs that can go and basically bind to that particular protein, say for example, which is involved in crosstalk with the human pathways and all. So this kind of information you can get. So the description of the model is consists of 75 components, in which 56 are host and 18 are bacterial virulence factors. The host components span molecular, cellular, and cellular state levels, while the virulence factors are specific to the bacterium. In the next slide, we have a table which basically lists the major immune responses. Integrate. Sorry. This is the table. So these are the events that are included in the model. So if you go like from first to last, this follows the trajectory. So entry into the host, then binding, then phagocytosis, then receptor mediated signaling, then phagolysosome formation, antigen presentation, T cell differentiation, immune cell influx, and then ultimately you do apoptosis and killing of the cell. So these are the different steps that have been modeled in this particular Boolean model. But there are other events occurring in this network as well, which are not considered here. But yeah, so these are the major events that are occurring. So if you're considering this, I think like good, and it is good enough. But obviously, you can give it more information. There are other stages of this infection as well, which you can use to model this basically host and host and bacterial infection by Excuse me. Okay. Yeah. So these are the different steps inside the after the infection, from the entry to the infection and apoptosis. So these are the different things that they are called. You can go through this. I'll not read these out because you can read it. These are simple information. Okay. So this is how the host pathogen network looks like. So this is you can see it's a very kind of what to say, like very dense and very like the network looks weird basically. So there are a lot of components. So first is these black arrows that are here. They indicate connection between processes that follow sequential. So these processes that I talked about, entry, binding, these things, they are shown by these black arrows. These green arrows here show activation, while the these red arrows, they show inactivation. Uh, and these blue arrows, they indicate signaling for proliferation or recruitment. And the various nodes have also been color coded. So the nodes that you see here. So the bacteria is indicated in darker colors, and while all immune cells here you see are colored in pink, and processes such as phagocytosis are colored in gray. So phagocytosis here, here also. So these are colored in gray, where the molecules are colored in cream, basically cream colors. And the cells NK, TH17, and GT cells. So NK here, TH17 is gamma delta cells. These are not basically, these are pronounced as gamma delta, not GT. So anyway, these are only implicated in vitro, and they have not been considered in the Boolean network. And all the boxes have been hashed. So basically, this is how the network looks like. So you can see a lot of interactions inside the network. There are activations, there are mutations, there are certain events, there are signaling entities also that are assumed inside the network. So this is kind of a very well detailed network that they have used. So how does the network look? So basically, so node representation, each node represents a component's qualitative state, which can be either on or off. So it can be 0 or 1. And all, then there is a Boolean transfer function. So transfer function, they'll utilize logical operators that can be and or not, etc. etc. So one example here is OR and NOT, or joint activation, which can be and inhibition, specifically and not. Uncertainty is handled by random variables, as we spoke about. So you just pick up with a probability, and you basically randomly assign a 0 or a 1, or kind of randomly assign a relationship between the two nodes if there is, or not if there isn't, but if you are not if you are unsure about the relationship. So then you can use random variables. In simulation, they have used asynchronous update, which is more biologically feasible, and the states evolved iteratively based on their update. So there are two. These are the two interaction events that I have listed here. These two interaction events have been modeled inside the network. So one is the reactive oxygen intermediates. So it is released by these APCs, which is known as antigen presenting cells, under pro-inflammatory cytokine influence. The bacterial defense components mitigate this ROI, ROI detrimental effect. So the Boolean function is basically, if you see, this ROI star is equal to activated phagocytic cells and pro-inflammatory cytokines, and not, and these are a set of genes given by or SODI or SODC or PPOB. So these are different elements. This can be genes or other elements as well. So the Boolean function is something that looks like this. This is how your function will look like. ROI star equal to activated phagocytic cells and pro-inflammatory cytokines, and not certain number of genes. Another example is the modeling of this TH1 cell that they've done. So these TH1 cells basically produce cytokines, which are basically IFN gamma and TNF alpha. These are cytokines, and these help in TB infection control. And these T cells differentiate into TH1 upon IL-12 or IL-18 simulation in the presence of bacteria. This macrophages increase this T cell production population in the presence of chemokines. So your Boolean function for this Th cell looks out something like this. So Th1 cell star equal to bacteria and Th cell and either of IL-12 or IL-18, and either this whole or macrophage and chemokines. So I just wanted to I have kept these two examples. Just wanted to show you how these Boolean functions are written and executed. Yeah. Okay. So next we come to the parameters of the model. So here I have described five parameters. There, I think they have used. I can see here 3, 6, 9, and 11. Sorry, not 9, 11, 10. 10 parameters they have used. I have described 5 here. So these are the most important parameters. So first comes the bacterial load. So B naught is given as the initial bacterial load, the minimum as the minimum bacterial load, maximum as the maximum bacterial load. And example variation is basically B0 can range from 1 to 100. Another parameter of the model is basically bacterial growth rate and its incremental change. So the growth rate will range from, these are like predetermined values, 0.17 to 0.59, and the incremental changes from, there can be incremental changes from 0.19 to 0.81, and it reflects the rate at which this NTB population increases. Another parameter is host clearance efficiency, which is given by Z. It represents the ability of the host cells to clear bacteria, and intake of drug can alter Z, which is a clearance rate. So here it is used as zero point 0.4. The other two parameters that are used is, first is delay factors for immune processes, which is given. There are two processes that they have used here. So DPH, which is the delay in phagocytosis, and DAP, which is the delay in apoptosis. So, delay in phagocytosis is the time taken for phagocytosis to achieve clearance, and delay in apoptosis is time for apoptosis to clear bacteria. So this you have to, they have assumed an approximately 7 step, each to account for gradual activation. So that they have incorporated inside the model. The other parameter they have used is onset delay of adaptive immunity. Represents the delay in adaptive immune response onset, and it is chosen as 14 time steps based on literature, and this can be adjusted for vaccinated individuals, which is, in which case DAI will be 0, because they already have the onset of adaptive immunity because they are already vaccinated. So I just I have listed down the parameters of the model just to give you an idea what kind of parameters they use. So, for example, here they have used bacterial load, how the bacterial load is changing. Also, they have used how the clearance is changing, meaning like after infection, how apoptosis and how phagocytosis is changing. Also, there is also a delay of adaptive immunity also, which they have modeled, because if you remember, like if you remember from your earlier studies. So basically, adaptive immunity takes time to kick on. So it'll maybe kick on after a day or two. After that, the adaptive immunity kicks on. So that there is always a delay between the infection and that adaptive immunity kick on. So that delay is also being modeled. So I just wanted to show you what kind of information they are using in the model as parameters. So just to give you an idea about how you can use it. So yeah, these are certain things that you can use in another models as well. Okay. Next is the simulation of this pathogenesis. So what they do is they initialize a initial set of components at infection onset, and the model evolution spans Tmax time steps, which is 105 in this case, 105 iterations, which is basically 105 in this case is the total number of time taken uh, in this case, so which is corresponding to 15 weeks. But the outcome is basically there are three uh, states that they have predicted, which is basically, first is the active disease, where the presence of bacteria is greater than the Bmax threshold. So the Bmax is here, maximum bacterial load. So if your presence of bacteria is greater than Bmax threshold, it is an active disease. Then, if there is no bacteria inside the system, then it is successful clearance. The last one is important because this is most common in humans, which is persistence. Bacteria is present within Bmax, within below Bmax only, or within Tmin or Tmax for a longer time. So what happens is, in this particular example, only TB only, in case of TB, so most of the people are unknown carriers, because those bacteria are in dormant stage, or they're not present above a certain load to create that infection, but they're carrying that infection. So this persistence is what is very common in case of tuberculosis, which they are very well modeled. So these are the different parameter values that they have used for simulation, and these are the results they've got. So active disease for 15 runs, clearance for 42 runs, and persistence for 43 runs out of the total 105 in this case. The extended simulation results are basically uh, parameters were used in 1200 runs, and which gave a result of basically 13% active disease, 41% clearance, and 46% persistent. So if you see the amount, like the probability that the bacteria will persist is quite high. That is the most common case actually. If you see in real world as well, persistence of tuberculosis bacteria within the host organism is very often seen without infecting the uh, person. So this is very well captured in this model. So you can say it's a good model. These are basically the other results from this simulation. So if you see, these are basically showing that this is what is known as the state map, and this is showing what are the active states. So this blue are representing the on states, and the white is representing the off state. So one thing, if you notice this state map, if you see in case of adaptive immune responses. So, for example, activated mast cells, activated neutrophils, activated T cells, or if you come down here, say Th1 cells, Th2 cells, TNF beta. So, these are elements that are associated with adaptive immunity. So, you see there is a certain period of delay before the activation of adaptive immunity goes on. So this is very well modeled here. So this highlights a delay in activation of adaptive immune responses machinery, and this delay may impact the progression and outcome of the infection. On the right side, you have the variation of bacterial load in systems. All the four plots are showing variations in bacterial load. So there are different conditions in here. Sorry. Huh, conditions here. So for C, in this case, the phagocytosis is disabled. So you see the bacterial load goes up to a certain saturation point and it stays there. If you see this one, D, uh, D is basically, I think, no delay in adaptive immunity. So if you have no delay, you are seeing delays here in adaptive immunity. If adaptive immunity kicks in immediately, so you have this spike, and after this, adaptive immunity also kicks in. So you have a complete like fall of the bacterial. This one is basically in which TGF beta is disabled. So TGF beta is basically a growth factor. In this case, if it is disabled, so if apparently if you do not have enough TGF beta, the bacterial growth falls down. So if you do not have basically if you delete TGF beta, knockout or something. So sorry. So the first one is basically for default parameter set. So this is how the bacterial load is changing with time, and these are the different perturbations. So this is if you do not have phagocytosis at all, and in uh, in in D, it's basically if the adaptive immunity kicks in immediately, and E is basically if you delete TGF beta. So what E is telling you is basically a drug target or basically a treatment strategy. With TGF beta, if you delete, there is a good chance that the infection will not persist. So these kind of inferences you can bring out of this model. So just to summarize the whole model, what they did was they took literature, they curated literature on host mycobacterium interactions. They identified components and interactions, 75 nodes, bacteria having 18 and host having 56 components. And along with that, 11 cellular processes, 19 cell states, and 26 molecules are modeled. Then you do the network assembly, and then you derive the Boolean transfer functions, and after that you simulate. So for simulation, you need the initial state node states. You initialize that set of nodes, and then you do the simulation based on your transfer function, and you'll have an outcome decision. And the outcome decision they have categorized in three categories. So if B max, so BT here is a threshold of basically bacterial load. If B minimum is greater than this threshold, then there is bacterial clearance. If the threshold is greater than the B max, then there is active disease, and something in between is basically persistence. So This was the whole model. Next, we come to the concept of robustness in biological systems. Uh, Intuitively, everyone kind of knows what robustness is like. It's basically the ability of a system to stay in its state irrespective of all the perturbations it is going through. So, and this is a property that is very often seen in biological systems, because biological systems have evolved over time, and with time, with evolution, they have like generate what to say, not generated. They have built this robustness inside them. We'll see what are the mechanisms through which the robustness is portrayed in biological systems. But yeah, this is a result of evolution, you can see. So, robustness is an ubiquitously observed property of biological systems. It is considered to be a fundamental feature of complex evolvable systems, and it is attained by several underlying principles that are universal to both biological systems and also sophisticated engineering systems. The only difference is, in sophisticated engineering systems, the thing is, you do not want emergent properties from sophisticated engineering systems, right? You want the system to work as you have designed. But in biological systems, it is kind of the opposite. There are emergent properties that you can see inside the network depending on the regulation of the network. And robustness can basically meaning, like, you can see robustness much more in case of biological systems than any other kind of system, because with evolution there with evolution the system has evolved in such a way that it can handle the environmental perturbations. This is the whole like idea of robustness and why robustness is there. So if you do not have it, because we live in such a complex environment, the environmental factors play a big role in basically how your genes get expressed, how your in general how your cells uh, express genes, express proteins, or how they go about their functions. Environmental factors play a very important role. So biological systems have grown to be robust, surviving in these environments, evolving over time. And this robustness facilitates evolvability, and robust traits are often selected by evolution. This is because this is why we have such a robust system after millions and millions of such robust biological systems after millions and millions of years of evolution. Such a mutually beneficial process is made possible by special architectural features observed in robust systems, but there are trade-offs between robustness, fragility, performance, and resource demands, which explain system behavior, including the patterns of failure. And insights into this inherent properties of robustness of robust systems, this provide us with a better understanding of complex diseases, and this can also be used as a guiding principle for therapy design. So on the right, this picture, Sir has also shown this picture in his videos. So this is a very good example of showing robustness. So the state of a system can be shown as a point in this state space. A state space. In this case, the state space is simplified to two dimensions, obviously, and the perturbations forcefully move the point representing the system state. So basically, this is your uh, attractor, and here is the cell, and here is the state, and due to robust adaptation, it returns to a periodic attractor. So this is the whole idea of robustness. If you perturb the system somehow, the system should work. The system should work regardless. So uh, the perturbations forcefully move the point representing the states. So basically, your after perturbation, the uh, attractor is moved to a new space, a new point in space, and the state of the system might return to its original attractor by adapting to perturbations. There are basins of attractions inside the state space within which the state of the system moves back to that attractor. So if there's a space within this, there's a kind of a space within the whole space. There's a space within the whole space in which if it is perturbed, it it can easily go back to the attractor. And if that boundary is exceeded, if that space is exceeded, on it, the state is not in that space, then it is the system might move into an unstable region or move to other attractors as well. So robustness is there in biological systems, but is there to a limit. There's a range, obviously. If you go above, very above, or very below that, the system will not stay robust. It is robust for a certain range. It is not robust for all and everything. So just keep that in mind. Okay. So next we look into mechanisms behind robustness of biological systems. So first is system control using feedback. So system control in biological regulatory networks, such as those governing cell cycles, circadian clock, and chemotaxis, rely both on positive and negative feedback mechanisms to achieve this robust dynamic response. The negative feedback, particularly integral feedback, is a fundamental mode of control that enables robust adaptation to the perturbations as exemplified in bacterial chemotaxis. Integral feedback ensures perfect adaptation by maintaining uh, responsiveness to a wide range of stimuli across the bacterial species. So uh, basically, you the system after perturbation, say for example, the system has a certain sort of a feedback mechanism to let the other components of the system know that there has been some sort of perturbation, and they need to adapt in a certain way. So those kind of signals are given by these feedback loops. So in most cases, you will see negative feedback loops because uh, in most cases it will be negative feedback because you'll need to stop. In most cases, you'll need to stop that production or stop the expression of the gene or something else. But in some cases, what happens in positive feedback is it amplifies the stimuli, which leads to bistability and a robust pattern formation as observed in this Drosophila melanogaster segment polarity formation. So in case of Drosophila basically development, there is in Drosophila development, after the segments of like basically after the embryo is formed, then the embryo goes into the anterior, the embryo, like what to say, defines it and its anterior and posterior side, and after that the segmentation occurs. Because these are like arthropods, so there will be segments on the embryo. So the segmentation occurs, and the polarity is uh, uh, it's very well maintained in case of the segments knows which is anterior, which is posterior. So these kind of uh, signals are generally positive feedback signals which amplify the stimuli and which lead to a bistability. So it can either be at a zero state or a one state. So your curve will look like something like a sigmoidal function. You'll have either a zero or a one state. So the idea is your negative feedback in its mode of control, which will enable robust adaptation to perturbation, and your positive feedback will amplify your stimuli, and it will lead to bistability and robust pattern formation, which is basically zero. And computational models have demonstrated that positive feedback loops are essential for reproducing experimentally observed patterns and ensuring robustness in biological systems. And the positive feedback also plays critical role in signal transduction, cell cycle, and fate decision processes, contributing to switch-like behavior. So anything you see a switch, so either a 1 or a 0 or a 0 or a 1, it's most probably a positive feedback. So this is something you can, next time if you see something like this, you can infer. So it, it contributes to a switch-like behavior, and it enhances robustness against noise and fluctuations in the stimuli. The combination of negative and positive feedback mechanisms allow biological systems to perform their functions effectively while maintaining the robustness. So one method that we learned how we can maintain robustness is through feedback mechanisms, either positive or negative. Next is basically the fail-safe option, which is redundancy and diversity. The concept of alternative or fail-safe mechanisms enhances the robustness by providing multiple means to achieve a specific function. So, the most common example would be isocymes. Uh, two different type of two different enzymes catalyzing the same reaction. So, it's basically if one of the enzymes will knock out, the other isozyme will take over, which is basically the alternative or the fail-safe. So this mechanism encompasses redundancy, diversity, and overlapping functions, offering flexibility and adaptability. While redundancy involves identical or similar components replacing each other, excuse me, diversity relies on heterogeneous components with overlapping functions. Uh, Sorry. Yeah, gene duplication followed by specialization and extensive gene loss serves as a crucial mechanism for evolutionary innovation. So gene duplication is another example of redundancy. So if you have multiple copies of the same gene, so even if one of those genes is mutated, it is very possible that the other genes will take over. So this what you can term as basically uh, uh, redundancy. This is not biodiversity. This is redundancy. But one thing you need to keep in mind that it's not always good for the system. So, for example, say gene duplication. So you can have duplication inside the cell naturally, inside your DNA naturally. But if it was so helpful in case of in preserving the robustness, then all genes should be duplicated. All genes should have two or three copies. But it is not the case because what happens is the regulation becomes much more difficult. And these type of duplications, this very big duplications that we have, say for example in Huntington's disease, we see a very small uh, gene that is getting duplicated multiple times, but that is going on to make a disease. So not all uh, like all mechanisms that are uh, promoting this robustness, not all of them are very beneficial for the cell. So the cell chooses which one to do when, but the point is not all of them are perfect for the cell. There are trade-offs. That is what I'm trying to say. At a network level, examples like glycolysis and oxidative phosphorylation demonstrate alternative pathways adapting to environmental changes, showcasing phenotypic plasticity as a part of robustness. So this is one example. Is basically aerobic and anaerobic respiration. So there are certain bacteria which, depending upon that environment, goes on to do aerobic respiration. When there is not enough oxygen present in the environment, they do anaerobic respiration for their energy needs. So these kind of adaptations, this kind of diversity, you can say, uh, uh, between alternate pathways or alternate mechanisms, which lead to adapting to the environmental changes, and these show phenotypic plasticity as part of robustness. Although redundancy is common in duplicated genes, duplicated circuits are rarely observed, suggesting that similar network topologies result from convergent evolution rather than duplication. So, as I said, like not all of them are perfect for the cell. So, redundancy, although common in duplicated genes, but you'll never see duplicated circuits because all genes won't be duplicated. Similar network topologies result from convergent evolution. They're moving towards the same state, all kind of convergent is basically all different elements moving to a same state, you could say. So this is basically the result of convergent evolution and not gene duplication. Uh, what they're trying to say. System control is vital to ensure robustness, as I said, for regulatory mechanisms, maintaining regulatory feedback and orchestrating switches between alternative mechanisms to preserve the system failure. So here is one example. In budding yeast, there are two genes, this CLB5 and CLB6. They encode B-type cyclins with overlapping function in cell cycle entry, illustrating the redundancy through gene duplication. However, the duplicated circuits are not reported, indicating convergent evolution rather than duplication. Phenotypic plasticity, such as diauxic shift. I don't know how much you know about diauxic shift. So basically, it's about shifting between the carbons, shifting of the state of the bacteria between the carbon source. It can be either glucose or galactose or lactose. So these are basically what's known as diauxic shift, which is very commonly seen in yeast, depending on the environment they shift, and which further contributes to robustness, allowing adaptation to changing environments. Next, uh, these are the last two concepts. These are the last two like mechanisms that I've described for robustness. There are other mechanisms as well, but these are the major. So the one here is basically modularity. Is a we learned about modularity in graph while doing basically the basic concepts of graph theory. So in there, the concept of modularity is a set of nodes inside the network which are more connected uh, in the network than it would be connected in any random network. So it is kind of a hub, a module, just making a module. So a similar concept is applied here. So what happens is the functions are module. So the function of a particular, say for example, glycolysis. So glycolysis will have a separate module, oxidative phosphorylation will have a separate module, which are interconnected much more than other modules with each other. So it encompasses physical, functional, spatial, and temporal modules, each contributing to the system's robustness and versatility. Examples include cells that form basic units of multicellular organisms and interact with the environment and other cells as well, hierarchically organized structures such as tissues and organs. Moreover, function modules within the cell, such as metabolic networks and signal transduction pathways, play a crucial role in regulating cellular processes. Logical modules, though less overt, are equally significant as demonstrated by systems like segmental polarity networks. The bacterial flagellum and its control module exemplify both physical and logical modularity, highlighting the adaptability and resilience afforded by modular organization in biological systems. So basically, it's you're compartmentalizing your functions. So it is very hard to quarter. So which will help you increase the robustness of this. So yeah, the concept of decoupling. Next is the concept of decoupling is serves to isolate low-level variations from high-level functionalities, thereby providing robustness amidst perturbations. Examples such as molecular chaperones like HSP90. This is basically heat shock protein. Fun fact: I think 2018. I don't remember. 2018, I think. 2018 Nobel Prize for medicine was awarded for heat shock proteins. Anyway, so molecular chaperones like HSP are which are not correct, which not only correct misfolded proteins but also buffer genetic variations, allowing for adaptation while maintaining this genetic diversity. So they perform two types of they correct for misfolded proteins but also buffer genetic variations. So it's kind of a two-way thing that it is doing. And feedback controls, as seen in bacterial chemotaxis and Drosophila segmentation, compensate for changes in network interaction or initial states, mitigating the impact of mutations. So, as we said, feedback control. And also bistability is created through positive mechanism and can decouple molecular fluctuations from the system's committed state, enhancing robustness to genetic and environmental correlations. So what happens is in case of bistability, it will decouple the molecular fluctuations with the system's fluctuations. So in case of bistability, the molecule can go from 0 to 1, thus the system goes to 0. That is what it describes. Yeah. Okay. So to wrap this whole thing up, this is a very good analogy to understand what robustness is. So, uh, modern day flights are designed, and there's some automatic, uh, what to say, automatic controllers within the flight, which are known as basically automatic flight control systems. And this AFCS, they maintain the flight's path against atmospheric disturbances by automatically correcting deviations from defined path. And it achieves this through feedback control, utilizing aircraft's flight control surfaces and propulsion systems. So this is what is known as the autopilot, what we call as autopilot. That is what it does is basically it maintains the flight path against all atmospheric disturbances. So these can be thought as perturbations, these atmospheric disturbances, and uh, going on the path, you can if you are able to go on that particular path without deviation, then you can say it's robust. The analogy that carries on. So yeah, it is achieved through these feedback controls. So there are sensors in various parts of the flight which will tell you different things, which is the wind speed, which direction are you going, what is the weather outside, everything. So using this information, it creates a feedback loop and directs the flight accordingly. And this robustness is ensured by incorporating redundancy and heterogeneity in this autopilot design. The redundancy is achieved by employing three modules of identical functions, ensuring that failure in one module does not compromise the system's performance. Similar we see in biology. Heterogeneity is introduced by designing each module differently to prevent common module failures. So modules are different. The concept of modularity in biological systems. This is kind of analogous to that. And digital technologies are utilized to implement this mechanism, decoupling low-level voltages from digital signals to prevent noise from interfering system functions. Here also decoupling. And while this example pertains to man-made systems, the principles of robustness extend to biological systems, highlighting the fundamental organizational principle of evolving dynamic systems across various domains, including evolution, competition, market needs, and society. So this is kind of everywhere, not in biology. Robustness is something that you see everywhere, and from an engineer's perspective, if you want to build a system, you want it to be as robust as possible. So okay, okay. So this is one of the architect. So robustness will have a certain architecture, right? So this is one of the architectures of robust networks. This is known as the bow tie. So it is the name is coming from the point from the fact that it's looking like a bow tie. So the network will look something like this, like a bow tie. You see here also, here also. So all these are different networks, but it looks like a bow tie. So what is it? It's basically a bow tie architecture will have many inputs that are fed from a central core that is the unutilized that is the unutilized to produce many distinct outputs. It's believed to contribute to the robustness of many biological methods. So the architecture is you'll have a lot of inputs. You'll have in the middle there will be a central core which will be producing different outputs, multiple outputs. So this kind of a bow tie architecture is very has been tested to be very good for the robustness of the system. In metabolism, the bow tie architecture provides a formal description of the large fan-in, as in the large input modules of the catabolized nutrients that produce a small number of activated carriers. For example, ADP or ADDH or ATP, and precursor molecules, which is basically the knot of the bow tie. Just before the output, that's the knot. So and basically so the elements are your input, which is the fan in the nutrients here. So this is the input, this is the precursor, which is the small core module, and after that it will produce different outputs. So this structure is what they are referring to. So the outputs they are calling as fan out, the input there is calling as fan in, and the middle they're calling the knot of the boot. This architecture provides exceptional robustness towards variable nutrient inputs through many degenerate metabolic pathways that can be used to produce a few building blocks, few common building blocks. A simple example of this degeneracy is glucose metabolism. It can take place either through glycolysis or through the pentose phosphate pathway. Although these pathways can substitute for each other if necessary, the sum of the metabolic effects is not the same. It's not the same of as like when it is working together, because like the cell knows when to use glycolysis and when to use a pentose phosphate pathway. It knows there are other elements also involved in that. So the requirement of those particular intermediates is depending on that, the cell will go through either glycolysis or glucose pentose phosphate pathway, but its metabolic effects will not be the same. But it is a redundancy within the system. It can be in the system. Okay, so here comes the end of the lecture. We have some MCQs to solve. So do it as we do. I'll put it on the screen and give the answer either in chat, or you can open your mic and tell me the answer. Whichever you prefer. Answer is correct. It's 2 to the 5. So it's basically 2 to the power n number of states that you can have, and n is the number of variables. Yeah. I'll just go back. So you guys solve this. Okay. So yeah, answers is correct. I'll just explain it. So this was the question. So G3 is expressed only when both G1 and G2 are depressed. It's not true. It can express when G2 is expressing as well. G3 is never expressed if G2 equal to 1 at all instance of time. It is also not true because it is dependent on sorry, G2 is never expressed if G1, yeah, G2, G3 is never expressed if G2 equal to 1 at all instances of time, which is not true. It is dependent on the value of G1 as well. It's not only dependent on G2, it's also dependent on G1. And if G2 equal to 0 in all instances, G3 is co-expressed with G1, it's not true because uh, G3 is can never be co-expressed with G1 because G3 is sorry G1 I think G2 G3 is not co-expressed with G1 because G2 if G2 equal to 0 G1 is not G1, so if G1 is expressed G3 will not be expressed, right, because G1 is not G1. And if G1 equal to G2, the next state of G3, basically G1 and G2 both are 1. So either of them is 1, you'll get G3. So good enough. It will be 1. It's true. It's just written in the opposite of it. So yeah, um, in the amount of inference you can get in Boolean is very low because you're using a lot of information when you're just considering 0 and 1 states. And stochastic. Oh no, sorry. Yeah, yeah. Did you answer? I just got confused.
No, it just it's the reverse order. Same thing. Boolean will have the
Same
highest.
thing.
Yeah.
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 It's the same thing. It's a reverse order. Yeah. So yeah, your stochastic simulation will have basically the lowest, or that will have OD, then you'll have RFPA because you're including those regulatory elements in the game. And Boolean, basically in terms of inferences, you'll get, but in terms of dynamics, is the next question. ODE will give you the maximum in terms of dynamics. Then RAPL will give you the highest inference, but like in terms of dynamics, it will give you nothing. In ODE, you will get the best dynamics possible. And then RFQ.
Is it redundancy?
It's redundancy. Yeah, it's redundancy. If there is similar type of genes doing, sorry, different types of genes doing similar kind of work, that you can classify it as diversity. But in this case, it's redundancy. Yeah. Just duplication, redundancy.
So negative feedback will be noise elimination, and positive feedback will
Uh,
be vice
that's
different.
correct. So in positive feedback, you will kind of have a sigmoidal curve on and off state.
Modularity.
These are straightforward questions.
Is it only C?
It's the opposite. Uh,
Oh,
so, so basically, the whole idea is, yeah, the whole idea is you'll have a set of input modules, you'll have a set of output modules, and in between you'll have a set of core modules. So in these core modules will be the precursors of this output. So your sugar, amino acid, everything will be formed from these precursor molecules. So there's a long list of input, and all of them are ultimately passing through that core module, generating these precursor molecules, and in their output they're giving the particular proteins, genes, whatever it is. So this whole thing, the structure, why is it so robust? Because different inputs are coming together to make the same set of precursors, and the outputs will be different for each input, but the set of precursors will be very same. Like if the inputs are quite different, then the outputs will be very different, but if it is kind of similar, the outputs will also be similar. So what it is trying is, what it is doing is, it is taking a lot of inputs and it is taking them to a intermediate stage where it is like producing these precursor molecules, which is like common for all of them. And then after that, the differentiation is happening and getting the outputs. So this is what is bringing in the robustness. So from all the set of input molecules, if you delete a few, there will still be precursors formed, and particular amino acids or sugars can still be formed because all the inputs are going through that precursor stage and forming that particular compounds. Are you getting me? So this, the whole architecture is such that, like, a lot of inputs, because you have a lot of inputs and you have the same set of precursors, it'll help you with the robustness. If you, even if a few of the inputs is gone, the precursors will still be formed and the outputs can still be formed. Get it?
okay, okay, okay. Yeah, yeah.
So that's the whole idea of the what to say, the bow tie idea. So
Mm.
this whole constriction thing is it's because you're taking all your input and you're constricting it to a same set of precursors, and the
Yeah,
output is different. Yeah. So that's all for today. So this was the, I think, the second last class. Next week we'll have the last class. So if you have anything you want me to discuss for the last class, because in the last class there are very few topics to discuss. So if you have anything in your mind that you want me to go through again, or if you have any doubts or anything, just you have my email ID. You mail it to me.
yeah,
I'll
yeah,
prepare the slides accordingly. So I'll just say, like, if you can mail it by this Friday, that will be good
yeah,
to have.
yeah.
Okay. So yeah. Thank you.
Yeah,
So
thank you.
see you next week.